Hello guys and welcome back to Excel Academy. So as part of protein sorting and transport, we were discussing like how proteins are transported from ER once they get incorporated into that, right? So now in today's topic, we'll be discussing about the Golgi apparatus. So we know that from ER, the proteins are then sent to the Golgi apparatus for the further modification. So organization of the Golgi, needless to say, I believe everyone knows about it. So we have got in the case of the Golgi, this is nucleus ER. So we have got Golgi stacks over here. And we know that it has got the cis, trans and intermediate compartments, right? So it is actually composed of flattened membrane enclosed sacs called cisternae and the associated vesicles. So Golgi apparatus is having polarity in both in structure as well as function. So what does it mean? It means that the cis part of your Golgi is in continuous give and take with the ER, whereas trans is having the protein sorting with the plasma membrane. So this defined about its polarity. Okay. Now proteins from the ER enter its cis phase, which is usually oriented towards the nucleus, this region. And then we have got the trans, which is facing towards the plasma membrane. Correct. So this is just the diagram. So here you can see this is in the case of the electron micrograph. You can see this portion is your cis, the intermediate Golgi apparatus we have, and then we have got the trans, which is facing towards the plasma membrane. Similarly, over here, the cis region is for your ER, medial, or the intermediate we say. Then the trans region is then trans-Golgi network regions, from where most of the vesicles are formed and which is meant to transport the proteins to the plasma membrane, or which is meant for the secretion, or the vesicle fuses with the endosomes or lysosomes. Okay. Now, again, the same thing, the Golgi consists of 4 discrete compartments, cis, medial, trans, and the trans-Golgi network. Now, what is important over here is that the sequence it follows. Okay. By mistake, if suppose sometimes the proteins which is carried into the vesicle from the ER and instead of joining to the cis region, if suppose by mistake it goes towards the trans, so what will happen? Golgi is meant for the modification. So that protein will be unmodified, right? And it will not be completely functional. Hence, then that protein will be targeted for the degradation. Okay. So this ordered sequence is very, very important for each protein to pass through. So proteins from the ER, Golgi intermediate compartment, they enter this cis compartment of the Golgi apparatus where modification of proteins, lipids, polysaccharides begins. Okay. After process through the medial and the trans compartments where further modification takes place, they move to the trans-Golgi network, jahan se, which is meant to distribute to the different organelles. So the transport vesicle associated with the Golgi apparatus in function to return Golgi resident proteins back to earlier Golgi compartments. Now, why these transport vesicles are coming back and binding to the Golgi compartments? Can anyone tell me why the empty vesicles comes and again go back and binds to the Golgi? Shruti, why you think they are doing like this? Is the question clear to you?
Ma'am, can you repeat once more?
See, in the cell, you have got the nucleus here. Here ER is there, let's say. Then this is cis, medial region, then trans region is there, okay? What I'm saying is that ER to cis region, it is happening, the vesicles coming and going. Now from the trans, the vesicles goes, let's say it is resulting in the secretion of some protein. Okay. Now what happens is that after some time, the empty vesicle which forms like this, they go and joins the trans-Golgi. So why the empty vesicles are formed and they go combined with the trans-Golgi area?
Because they can be recycled and used.
Yes, absolutely correct. Otherwise, what will happen if this continues? The Golgi trans region will become shortened, right? And the plasma membrane for no reason will become more. The length will be more, correct? So we don't want that to happen. So in order to recycle or reuse the membrane, which was fused because of the Golgi, they take it back and go empty-handed. So most of the time it is not empty-handed. They do have the Golgi apparatus resident proteins, like how we have got the resident proteins of the ER, right? So if they have got those sequence of KDL and KKX, which we discussed yesterday or in the previous lecture, so those resident proteins will go back to the ER. Similarly, in the case of Golgi apparatus also, certain resident proteins are there. So to that, those vesicles will go back. Okay. What you need to understand here is that if suppose this is trans Golgi and this is plasma membrane, the vesicle which has formed, okay, it gets fused to this. Let's say it got increased this much. Okay, it does not mean that immediately after the secretion, this region will be pinched off and go back for the binding with the Golgi apparatus. Okay, it's not necessary that ki wahi region jayega. It just can be from the other area also, but having the resident proteins of the Golgi apparatus. Okay, did you get the point here? So every time when vesicles are released or secreted out, the protein samples are secreted out, it is not that only one vesicle is there, right? Itna sara cargo sirf ek vesicle mein nahi hoga, kam se kam honge 1000 vesicles, which immediately go and fuses with the plasma membrane. And if that particular region, let's say that these regions are having your resident Golgi apparatus proteins, so that particular region will pinch off again. Those vesicles will go and back and fuses with the Golgi apparatus trans region. Clear now?
Yes, ma'am. But I wanted to ask about the ER. So when a part of the ER gets pinched off and goes to the Golgi apparatus along with its resident proteins, doesn't that won't there be a difference between the environment of the ER, environment of the Golgi apparatus, environment of the cell membrane? All of those will be altered, right, when they exchange vesicles like that?
No, so that's what na. It is the composition of the lumen which is different, but the pH is same. Okay. So if suppose environment was different, then our protein, which is like translated in the cytoplasm, which goes for the modification in the lumen and in the Golgi, then these proteins will get altered if the pH is different. Correct? So the pH remains same. It's just that the location of the proteins which is meant to help the other proteins to get modified. Okay? Samjha? See,
But,
when you, if ER is there and Golgi is there. Okay? So you are saying that this pinch off, it goes and fuses with this. Correct? That's what you are saying,
yes,
right?
Yes,
the environment over here is going to be different from the environment over here. That's what you are saying?
Yeah.
Why, Shruti? Why the environment will be different? Only difference is that the proteins which is resident to the ER and the proteins which is resident to the Golgi apparatus is going to be different. And that's how, that is what determines that is ER and the other one is Golgi apparatus. Otherwise, the lipid composition is going to be almost same. The pH is going to be same.
Yeah, this part I understood, but with respect to the cell membrane, which is connected to the outside of the cell, over there will be a difference, right?
So again here the lipid composition is same. ये lipid cell membrane का कहाँ से आया? Just now we discussed, right, yesterday, that it is the SER which synthesizes the lipid, phospholipids. So that is this vesicle which carries the lipids. जो हमने extra synthesize किया है, it is actually in the ER, okay, of your SER. यहाँ पे रफ की जगह पे स्मूथ एंडोप्लाज्मिक रेटिकुलम में आपका लिपिड बन रहा है जो अपने ही मेम्ब्रेन में ऐसे ऐड करेगा. That particular thing will become a vesicle which go directly and fuses with the plasma membrane.
Okay.
Samjha? Now why do we need lipid synthesis? Because when a cell is dividing, it needs to enlarge itself, right? It needs to increase the size. So that time we need high rate of lipid synthesis. क्योंकि जब सेल डिवाइड हो जाएगा आधा उधर हो जाएगा between the two daughter cells, right? And we don't want like from here to having small cells. We want having a good or equal length of the cells, correct? So that time lipid synthesis are at high rate. Okay, now you got it? So what I was saying was that plasma membrane mein, if suppose these are your Golgi apparatus related resident proteins, okay, so this particular region will be understood and ye aapka endocytose hoga without anything. Okay, यहाँ पे आपका कार्गो कुछ नहीं होगा. It will be empty vesicle. This will go and bind to the trans-Golgi area, so that the length of the Golgi does not decrease and the length of the cell membrane does not increase during this vesicular trafficking. Okay. All good, Shruti?
Yes, ma'am. Yeah.
Okay. अरे क्या हुआ? Okay. So now coming to the protein glycosylation within the Golgi. So as I mentioned before also,
Ma'am, you stopped sharing the screen.
Oh, it got stopped, yeah. Now it is visible.
Yes, ma'am.
Okay. So yesterday we,
Like I have a doubt, like while this is getting recycled, like the vesicles again going into Golgi bodies, like what if it accidentally takes out any lipid from the membrane again inside? So will it be degraded?
No, lipid is what? Phospholipids, they are present in the Golgi membrane also. Same phospholipid is present in the membrane also, cell membrane. So why it is going to degrade? It's okay even if it takes 1 or 2 phospholipids extra.
Okay.
Okay, so yesterday in the previous lecture we have discussed like the glycosylation. So what kind of glycosylation happens in the ER? Anyone remembers? We talked about 14 sugar oligosaccharide. Do you remember that? So in the ER, it is your N-linked glycosylation. I told you to remember, right? In ER, N-linked. So that is asparagine में आपका bind होगा. Sorry, N it is N. Asparagine में it will bind N-linked. So 14 sugar क्या क्या है आपका? 2 N-acetyl glucosamine, 9 mannose, and 3 glucose. Right, of which we said that the 3 glucose gets removed on the further modification. Now from there is going to be different glycosylation or the modification happening inside the Golgi. Now in the Golgi you have got more than 250 enzymes which is going to have the different kind of modification requirement. So जो जैसा enzyme involve होगा वैसा वैसा आपका protein modify होगा, ठीक है? And all these enzymes are located in different compartments of the Golgi. So what does it mean? Cis में जो होगा वो आपका medial में नहीं होगा. Okay, so all these 3 regions are going to have different enzymes which is having their own specific modification role. Okay, so these enzymes are located in different compartments of the Golgi. So carbohydrate processing takes place in an ordered fashion as a glycoprotein travels through the Golgi complex. Okay, so different glycoproteins are modified in different ways during the passage from cis, middle, and trans. Sometimes there won't be any modification at the cis and middle, directly in the trans hoga, or whatever the combination required, okay, depending on the function of the enzyme or the protein. So here you can see, so mannose residues are removed and N-acetyl glucosamine is added. So this is that 14 sugar oligosaccharide which was added in the ER. So 3 glucose were removed in the ER itself, right? Now that protein is transported to this cis region of the Golgi where again the mannose are removed and N-acetyl glucosamine were added again. Or you can add fucose, N-acetyl glucosamine and galactose are again added. Or you can have the sialic acid added. So eventually what is happening? You are getting a type A or type B kind of, that antigen which is present on the cell. So here you can see that cis में आपका कितना modification हुआ, that mannose were removed, N-acetyl glucosamine was added. Middle region में क्या हुआ? Fucose and NAG was again added. Then trans में क्या हुआ? Sialic acid was added. So each step, that processing of all these oligosaccharide, okay, is important, which goes through or which happens in the Golgi. Okay. So this is a processing of your N-linked oligosaccharide in the Golgi. ये एक काम हो गया Golgi का. दूसरा काम क्या हुआ? Having its own O-linked glycosylation. ठीक है? This is same thing written over here. Now, we have got another type of glycosylation, and this one is really very important. This is something you have to remember. See, you see that here 2 NAG are there and 9 manos are there. Okay. After your N-linked oligosaccharide was added in the ER, this is what gets transported to the Golgi. Now, if suppose this protein is meant to be transported to the lysosome, इसका मतलब क्या ये lysosomal protein है, then after your 2 NAG and 9 manos, there is going to be another kind of modification. That is, यहाँ पे क्या होगा आपका phosphate group add हो रहा है. So UDP becomes your UMP. Q, because one of the phosphate group is added over here. Okay, did you get the difference? And later this, so this is removed. What is required is that at the 6th mannose, the phosphate group has been added. Now this becomes the tag. It is known as tag or lysosomal tag. So all the proteins which is present in the lysosome, they are going to have this mannose 6 phosphate का tag. Okay. Now this is like a signal. जैसे अभी हमने न्यूक्लियस में पढ़ा था NLS बोलते हैं, KDL हमने पढ़ा था ER के लिए, वैसे ही लाइसोसोम के लिए हो गया आपका mannose 6 phosphate. So wherever this mannose 6 phosphate protein is present, so the receptor or the others transporter proteins, they will recognize this tag and recruit this protein into or towards the lysosomes. Clear? Is it clear, guys?
Yes, ma'am.
Yeah. So this is important. Please do remember, this one is really very, very important. Okay. So same thing written over here. Now, so the phosphorylated mannose residues are specifically recognized by the mannose 6 phosphate receptor in the trans-Golgi network, which will direct the transport of these proteins to the endosomes or the lysosomes. ठीक है? तो इसका मतलब क्या हुआ? Mannose 6 phosphate तो protein आपका present है, which got modified in the trans-Golgi network. That will be recognized by the mannose 6 phosphate receptor, जो उसको lysosome तक पहुंचाएगा. ठीक है? Now coming to the second category, that is O-linked glycosylation. So again, these modification will take place in the Golgi. For example, the proteoglycans. अभी ये भी है जो आपको याद रखना होगा. So just now what we have discussed, let's summarize. ER में होता है आपका N-linked glycosylation, okay? जिसमें से हमने जो पढ़ा 2 NAG, 9 mannose, 3 glucose. So 3 glucose will be removed, 9 mannose modify होके आपको mannose 6 phosphate का tag देते हैं, which is for your lysosomes. ठीक है? This is one example. Another example where we have discussed where the NAGs were added, fucose was added, galactose were added. That is just to explain that the modification, different modification is happening in the different compartments of the Golgi. That is another function of your Golgi. Now, third function of your Golgi is the O-linked glycosylation, to carry out O-linked glycosylation. Now, where this is important, it is important for the formation of your proteoglycans. Now, what are these proteoglycans? So, they are nothing but part of the secretory proteins which forms your extracellular matrix. Okay, so they are the good example of your extensive O-glycosylation. So, their processing happens in the Golgi apparatus where a 100 to 200 sugar residues are getting added. Okay. Now let's see the structure of this. It's okay if you don't remember the structure, just try to understand. Okay. So here you can see there is a 1 polypeptide chain. Yes. N-terminal and C-terminal is there, right? So this is your main core protein, the main backbone protein is there. After that, what we see is that so many sugar molecules are being added, and those are all your modified sugars. Okay. Now, when I say modified, it means what? Like you don't have directly the N-acetyl galactosamine. Okay. It is your chondroitin sulfate or your keratin sulfate. So where do we hear all this about? So along with the collagen, they form your extracellular matrix. Okay, so here you have to remember that to your proteoglycan, you have got many or several copies of this chondroitin sulfate and the keratin sulfate. अब यहाँ पे याद क्या करना है for exam? Okay, that is what is main thing. So proteoglycans are nothing but they are the extracellular matrix components which is made up of modified glucose like chondroitin sulfate and keratin sulfate, where these chondroitin sulfates are made up of glucuronic acid and N-acetyl galactosamine. Please guys make a note that हमने इससे पहले जब हम N-linked glycosylation पढ़ रहे थे where there we have talked about NAG. ठीक है, तो N-acetyl glucosamine को हम NAG बोलेंगे. ठीक है, यहाँ पे हम NAG लिख सकते हैं but we don't write, we write it like this. Okay, short forms जो रहते हैं N-acetyl galactosamine, glucuronic acid, then keratin sulfate हो गया आपका NAG plus galactose. So this is something which you have to remember, और यही चीज रिपीट होके हमें बायोकेमिस्ट्री में दोबारा पढ़ना है. So it's better that you remember now itself so that we don't have to repeat it again. Is this part clear to you? Like where the Olin glycosylation is happening, what is the example, and this example में आप क्या बोल सकते हो? So the core protein is there which is made up of these modified sugars. Yes, clear? Any doubts so far? Krishita, any doubts over here?
No, ma'am.
Okay. Now coming to the lipids and the polysaccharide metabolism in the Golgi. Okay, so that is in the synthesis of the glycolipids and sphingomyelin. So अभी हमने क्या देखा था that in the ER, smooth ER, your phospholipids are getting synthesized. So yesterday we saw how from the glycerol, phosphatidic acid is present, it gets inserted between the phospholipids, and then how the different precursors when they go and bind. For example, choline हुआ तो phosphatidyl choline मिला, inositol हुआ तो phosphatidyl inositol मिला, right? So those were your phospholipids. Now about the glycolipids and the sphingomyelin. Okay, so the glycerol, phospholipids, cholesterol and ceramide, they all are synthesized in the ER, whereas sphingomyelin and glycolipids are synthesized in the Golgi apparatus. So sphingomyelin is synthesized from the transfer of a phosphatidylcholine group from a phosphatidylcholine to ceramide. तो यहाँ पे क्या होगा? What you have to remember is ceramide. Ceramide, when you add this group, you get a sphingomyelin. ठीक है? So इसका core क्या हुआ? इसका base क्या हुआ? आपका ceramide. Okay. Now, if you add different sugars, okay, different sugars to the ceramide, you are going to get different glycolipids. Let's see the structure. Don't worry, we have got the structure. Okay, so this is your ceramide. Now, can you see the difference between the ceramide? Do you remember like how glycerol looks? Can you see the difference? क्या है difference? Glycerol, हमने जो glycerol 3-phosphate हमने जो पढ़ा था and इसमें, so you can see over there the structures, we don't have this group in the glycerol. ठीक है? This difference is there, and later 2 fatty acids are attached in the phosphatidic acid also. Okay, so this group makes a huge difference in the ceramide. Now, when you add phosphorylcholine group, it becomes a sphingomyelin. If you add any sugar, it becomes a glycolipid. आगे जाके हम पढ़ेंगे cerebroside is there, then globoside is there. All those different types of glycolipids are present. So those are nothing but made on the base of your ceramide. ठीक है? Don't worry, कोई कुछ नहीं पूछेगा, just for your understanding. You don't have to study the structure here. Okay. Okay, this is something which we have to refer. The sphingomyelin. Am I going fast? Are you able to understand? Even if not remember, are you able to get the concept?
Yes, ma'am.
Okay.
Yes, ma'am.
Guys, whenever you get confused ना, जब भी आप इसको revise करोगे, keep in front of you the structures, okay? Even though you don't have to remember for the exam point of view, but at least for your understanding, okay? So sphingomyelin, they synthesize on the luminal surface of the Golgi, but glucose is added to ceramide on the cytosolic side. Are you getting over here? ये आपका cell हो गया, ये आपका Golgi हो गया, okay? गोल्जी के अंदर की तरफ आपका स्पिंगोमाइलिन बन रहा है, बट जो शुगर ऐड होता है वो आपके इस तरफ ऐड होगा जो आपका साइटोप्लाज्म में प्रेजेंट है, ठीक है। सो glucosylceramide then apparently flips. However, additional carbohydrates are added on the luminal side of the membrane. So glycolipids are not able to translocate across the Golgi membrane. So they are found only in the luminal half of the Golgi bilayer, as is sphingomyelin. अब आपको समझा स्पिंगोमाइलिन क्यों हमेशा हमें इंटीरियर लीफलेट में मिलता है ऑफ योर प्लाज्मा मेम्ब्रेन? It is because they are not able to be synthesized on the cytosolic side, right? So जब भी वो फ्यूज होगा तो inner membrane of your Golgi, inner membrane of plasma membrane is going to be the same then. Okay, so this concept will be clear once we study how the vesicle fuses with the cell membrane. Okay, I'll just try to explain now. I mean, I'm not that good in diagram. So they say this is the plasma membrane का outer and inner leaflet. Okay, let's say Golgi से आपका एक vesicle आ रहा है, उसका outer ये है, उसका inner ये है. So how it is going to get fused with this? तो यहाँ पे कट होके this portion outer should bind with the outer and inner should bind with the inner, right? Correct? So what happens is that there is a cut made over here. Then अब इसको कैसे बनाऊं मुझे भी नहीं आ रहा. What I want to make it is that ये ऐसे यहाँ join होगा ऐसे. Okay, I'll make it with this. Green वाला नीचे वाले को join होगा. So this gets open up like this. ऐसे. So whatever content is present in the vesicle will go out, and this is how it will look then later. Did you get little idea? Anyone? Tanvi? थोड़ा बहुत आइडिया आया कैसे एक वेसिकल प्लाज्मा मेम्ब्रेन में फ्यूज होगा?
Yeah, ma'am.
Yeah, I'll try to get animation. I'll just check out if I get this thing. Anyways, this concept will be taken in detail more later. Okay. Yeah.
Ma'am, I didn't understand the second
Second what?
line in the slide. Just a
So,
preview.
yeah. So Krishita, the thing is, I am not that good in diagram. I'll try again. Okay, I'll try again. So what point is that? ये plasma membrane का, this you understood when I say plasma membrane ka outer and inner, right? This
Yes,
you understand, huh?
Yes.
So whenever a vesicle is formed, it is like this. Even this is clear to you. यहाँ पे आपका cargo रहेगा, or whatever the enzymes which is meant for the release outside the cell, correct? So what I wanted to say is that outer will bind with the outer, ऐसे, when it gets open, and inner will bind with the inner membrane, ऐसे. You have to imagine everything in 3D. This is in 3D, okay? It is not 2D, it is 3D. So,
Ma'am, this I understood, but only sentence, I was a bit confused over that second sentence. Look, if you could just go back.
here,
No, ma'am, the previous one. This glucosylceramide, the next one.
where, which one?
The glucosylceramide then apparently flips. However, the additional carbohydrates are added on the luminal side of the membrane.
So,
That
okay,
is-
okay. See, this is not related to that, okay? The fusion which I was talking about, it is not related to that. This is completely different topic. So I was talking about sphingomyelin and glucosylceramide, right? So sphingomyelin means, one second. If this is your Golgi, तो ये आपके यहाँ सिंथेसाइज होके यहाँ बाइंड होगा. इट विल बी प्रेजेंट हियर. Okay, this is what I was saying. Sphingomyelin is synthesized on the luminal surface of the Golgi. So this means luminal surface. Golgi के अंदर का जो हुआ वो आपका luminal surface हुआ. Okay. Now glucose is ceramide then, but glucose is added to ceramide on the cytosolic side. So when ceramide is present, ceramide can be present here also. So sugar moieties will be added over here like this, because these enzymes are present in the cytoplasm. Itna clear hai?
Yes, yes.
Huh? Now what we are saying is, so I hope you know about the enzymes called flippase, floppase, scramblase, right?
Yes,
So they
yes.
help in the flipping of this glucosyl ceramide to this side. So kya hoga abhi? Let's say this is this. After with the help of the flippase, floppase, it will come over here, right?
Oh, okay.
आउटर से इनर की तरफ आएगा, यानी कि साइटोसोलिक साइड से ये लुमिनल सरफेस की तरफ आएगा, एंड लेटर द फर्दर मॉडिफिकेशंस विल बी डन. दैट इज व्हाट इज रिटन.
Okay, okay.
Okay, yeah. Now coming to the plants. Okay, so in case of plants, Golgi apparatus have got extra work to do apart from what we have just talked about. Okay, so it is involved in the carbohydrate modification, it is involved in the lipid synthesis, etc. Right? In the case of the plant, it is also required for the cell wall synthesis. So we know that this plant cell wall is made up of major polysaccharides like cellulose, right? So it is a simple linear polymer of glucose residue. So all these so it is synthesized at the cell surface by the enzymes in the plasma membrane. So you can imagine like how cellulose, hemicellulose, pectins, whatever it is present, they synthesize in the Golgi, and then they transport it in the vesicles to the cell surface, where with the help of these enzymes, they keep on adding to the cell wall. Did you get that thing? Able to imagine? I'll try. So this is the thing is that I don't have figures and all given. So let's say this is the plant cell wall. Then you have got the plant membrane. And I'm just making only Golgi here. Okay? So from Golgi, all these lipids, all those sugars, whatever is required for your cell wall synthesis, everything will be taken and put into the vesicles. ठीक है, ये vesicles जाएंगे आपके plasma membrane के पास. यहाँ पे आपके रहेंगे the enzymes required for your cell wall synthesis, which is attached to the cell membrane. Then they keep on adding over here. ऐसे add करते जाएंगे so that they can increase the length of the cell wall. अभी थोड़ा सा idea मिला. I didn't get any figure related to this, otherwise I would have added. If you guys see, let me also know, but I hope little idea you got, like little imagination, like how the things, the raw materials are taken from the Golgi, it goes to the cell membrane, and from cell membrane, they help in inserting into the cell wall. Yes? थोड़ा सा आइडिया, एनीवन? नो रिस्पॉन्स. Nobody is able to imagine. Akshaya?
I got it, ma'am, but are the enzymes present in the membrane of the cell?
Yes, peripheral proteins.
Okay.
Yeah, okay. Now coming to the protein sorting and export from the Golgi apparatus. Okay, so proteins as well as lipids, polysaccharide, they all are transported from the Golgi apparatus to their final destination through the secretory pathway. So what is the secretory pathway? Already we have discussed, right? जब आपका ट्रांसलेशन होने के बाद फ्रॉम ईआर टू गोल्जी, गोल्जी से फिर आपका वेसिकल बनता है, वेसिकल से टू द रेस्पेक्टिव ऑर्गेनल्स, आइदर इट इज लाइसोसोम्स, माइटोकॉन्ड्रिया, क्लोरोप्लास्ट और प्लाज्मा मेम्ब्रेन फॉर द फाइनल सेक्रीशन आउट. ठीक है, so I think I have a figure here. Yes, so let's see in the figure. So you have got over here trans-Golgi area, okay, jahan se aapka vesicle banega. So you can see that each compartment, okay, it is meant, or you can say that it is localized for types of proteins. Okay, for example, let us say that if these are the granule proteins, okay, granular proteins are there, so this particular thing will be immature. On the pathway they become matured, okay, in the vesicles and finally released. Or you can have a receptor associated proteins, so that will be taken, it goes and gets released. Right now here, if suppose there are certain proteins which is meant to fuse with the endosome, so they will go, they will bind to that and those will be released then. Or the proteins are meant to get fused with the lysosome, yani ki wo lysosome ke proteins hai, so they will go and bind to the receptor. Let us say that this is mannose-6 phosphate receptor, which recognizes the mannose-6 phosphate tagged proteins, that will be taken, fused to the late endosome or the lysosome, so that becomes your active lysosomes. Right, so these proteins are present at the pH of around 7, we know that, right? But we also know that in the lysosome the pH is less, around 3 to 4. So then only these proteins becomes active. Otherwise kya hoga, these lysosomal proteins are hydrolases by nature, yani ki unka kaam kya hai, to just digest and degrade, right? So since they are present in the neutral pH or optimum pH, that time they are inactive. The moment they reaches the lysosome and lysosome mein pH aapka kam rehta hai, that becomes active. Okay, so this is how from the trans-Golgi network to different destinations with the help of the vesicle they are being transported. This is known as secretory pathway. Clear? So I have got one video, let's watch to understand this more. One second. Let me share the audio also. Are you able to see the screen, that video?
No, ma'am.
Yeah.
It's still the slide.
No, ma'am.
Okay, I guess I need to share it again. Now?
Yes, ma'am.
Okay. I think this is just a video which shows, this is for budding of a secretory cargo from a Golgi apparatus, okay, like how the vesicles are being formed. This is in real time. Wow. Yes. Did you get how the vesicles are being formed and then again they come back and join? Were you able to understand? This is like real time. I'll show once again. See, here you can see few things are like pinching out and going. So those are your vesicles if you see very carefully. And then few from the back end, they're coming and the vesicles joins back. See, this one again got pinch out. This one got pinch out. It is going, it is getting coming back. It is going. It gets modified, it does its work. And then later you will see that it comes back and joins. Yeah. Yes. Thoda idea mera, like in real time it will look like this. Okay, so they have used, I don't know, the fluorescence and they have taken the real time video of this. Okay, now there is one more which I would like to show you. Yeah, after the post Golgi transport. Okay, let's watch this also. So many vesicles are there, so many. Yes. Guys, did you see? Were you able to understand? Tanvi? Krishita, were you able to understand what exactly was happening in the video?
Yes, ma'am.
Okay, good. So you must have noticed like there was ऐसे चींटी जैसे ऐसे बहुत सारे छोटे so many were there, right? So since they have shown in very real time seconds, that's why it was so fast. So many small vesicles coming and going, coming and going. So I'll just show once again and then we can proceed. So there were many are there, many coming and going, coming and going. So when all this vesicle transport is happening, even though it is so fast, you need to understand that this all is happening very fast and each step, okay, I'll tell you one thing. See, ये अगर आपका ट्रांस गोल्जी है, ठीक है, यहाँ से आपका वेसिकल बन के जा रहा है, okay, it is going to the plasma membrane. Let's say this is your plasma membrane for the fusion. So it's not that the vesicles are getting transported to the plasma membrane simply. ऐसा नहीं है. It is not floating in the cytoplasm. No. जैसे हमारे कार्गो के लिए एक व्हीकल रहता है, let's say some truck is there, right? So from one destination to another destination, the truck is traveling on the road, right? In the same way, inside the cell where all these organelles are present, everywhere the network of actin filaments is there. The network of microtubules are there. So ये क्या करते हैं? So we have got roads like this. Microtubules ऐसे present रहते हैं, criss cross. And जो कहते हैं ना, जो कच्ची गलियां रहती हैं, जो जैसे हमारे एक हाईवे रहता है, एक आपका सर्विस रोड रहता है, या एक छोटे छोटे कच्चे पाथ रहते हैं. Imagine those are your actin and highway is your microtubules. So on these microtubules, each carrier proteins are there. Later we'll talk about them. Those are your dynein and kinesin. सुना भी होगा. These are your microtubules motor proteins. ठीक है? So these proteins, they carry this cargo. Okay, they carry like this. और एक एक स्टेप जब वो रखते हैं, एक दो, जैसे हम चलते हैं, सेम वे इवन दे वॉक, ऐसे ऐसे एक कदम आगे. So each step needs ATP. So right now you saw that there were so many carriers here and there they were going, right? So just imagine one step means one ATP. So how much energy is being utilized? So that's how from Golgi to the plasma membrane, this vesicles are being transported here. और अगर कोई चीज endosomes बनके या endocytosis होके अंदर की तरफ आता है, so that is also being carried out like this. ठीक है? It exactly looks as if a person is holding a cargo on his head and walking. वो वैसा ही दिखता है. I'll show you once we talk about these motor proteins. Beautiful animation is available. Okay. Is this much clear to everyone? Yeah.
Yes,
Now this is also a part of Golgi only. So in the case of the epithelial cells, so if you know the epithelial cells, एक part होता है that it is going to absorb the nutrition, and one part of your epithelial cell is which is taking this absorbed nutrition and sending into the blood circulation so that it will be transported to all different regions of your body, right? तो उसका जो what you say, the polarization we say, okay, it is different. अगर ये आपका epithelial cell ऐसा है, so the proteins present over here is going to be different from the proteins present over here. Okay, those are the apical region and the basolateral region. So the receptors or the proteins localization present here is different and here it is different. So Golgi apparatus has to be very specific. It needs to understand कि एपिकल मेम्ब्रेन ऑफ द इंटेस्टाइनल एपिथेलियल मेम्ब्रेन में आपको क्या भेजना है और आपको बेसोलैटरल रीजन में क्या भेजना है. ठीक है. So that is very specific job of your Golgi and it has to be very selective. Okay. So the plasma membrane of such cells, like for example, in the case of epithelial cells, they are known as polarized. So they are divided into two separate regions, the apical and the basolateral domain, which contains specific proteins. For example, apical membrane of your intestinal epithelial cell faces the lumen of the intestine, which is specialized for the absorption of nutrients, whereas the remainder of the cell is covered by the basolateral membrane. Thus, proteins leaving the trans-Golgi network must be selectively transported to these distant regions. Otherwise, what will happen? आपका nutrition अच्छे से absorb नहीं होगा and passing to the different areas or different in the blood circulation वो भी आपका अच्छे से नहीं होगा. ठीक है. Let us say about this. So consider one epithelial cell. So you can see here microvilli are there. So these microvilli are coming under the apical region. Okay. So they are meant for the absorption of the nutrition. Okay. Now, if this is meant for the absorption of the nutrition, then here whatever is present, okay, जो भी आपने खाया पिया, it should be converted into glucose or whatever. Then it should be transported into the basal lamina, जहाँ पे नीचे आपके क्या आएगा? यहाँ पे आपका endothelial cells आएगा, or you can see that blood vessels आएगा इसके नीचे. So that the glucose will be taken and transported to the different regions of your tissue, so that वहां पे ग्लूकोज एब्जॉर्ब हो एंड वो अपना टिश्यू अपना काम करे. ठीक है. सो गोल्जी ट्रांस गोल्जी हैज टू बी वेरी स्पेसिफिक कि एपिकल रीजन में कौन से प्रोटीन भेजने हैं और आपको बेसोलैटरल रीजन में कौन से प्रोटीन्स है, इंटीग्रल प्रोटीन्स हैव टू बी स्पेसिफिक. ठीक है. वो कौन से भेजने हैं. Did you get it? Now how it is being done? For this also they need certain signal. Okay. So this is accomplished by the selective packaging of the proteins into the transport vesicles, which is targeted either for the apical or for the basal. Okay, which make this takes place in the trans-Golgi network only. For example, with the proteins which are targeted to the basolateral domain are they are the short amino acid sequences such as dileucin or having tyrosine containing hydrophobic motifs. Okay. Did you get it? For basolateral, you have got either double leucin or tyrosine containing hydrophobic motifs. So now they are meant for your basolateral specific proteins. Now, if it is your apical specific, then they should have signals consisting of your GPI anchors. Okay, rather than having an amino acid sequence, they should have a GPI anchors or any carbohydrate modification. So this is how the trans-Golgi network understands that कि मुझे apical में भेजना है या basolateral में भेजना है. Okay. Yes. So any doubt, guys? I'll be stopping here then.